Welcome to The Explainer. We're all about turning complex research into clear, dynamic presentations.
Today, we're basically becoming scientific detectives. We'll start with the big mystery of the bipolar brain, then we'll hunt for clues in brain scans, and after that, we'll zoom way in, under the microscope. From there, we'll see how it all connects, and finally, we'll look at where the search stands today. Okay, so let's jump right in. Every great mystery story has a central challenge, right? Well, when it comes to bipolar disorder, the big one is this. Why is it so incredibly difficult to find a clear, physical fingerprint of the illness inside the brain? Let's dig into that.
So, let's stick with this detective analogy. If you look at a disease like Alzheimer's, it's like finding a smoking gun at a crime scene. Pathologists can actually look at brain tissue and see these very specific things, protein plaques, tangles, they're right there. But with bipolar disorder, it's like the crime scene has been wiped clean. There's no single obvious clue that lets a researcher point and say, aha, that's what we've been looking for.
And that, right there, is the heart of the problem. That's the challenge. The scientific term for these clues is biomarkers. And without robust, repeatable ones, everything gets so much harder. How do you make a rock-solid diagnosis? How do you track if the illness is progressing? And maybe, most importantly, how do you develop new treatments that get to the real root of the problem? This lack of a clear target is exactly why the search is so important.
Okay, so even if there's no smoking gun, it doesn't mean there are no clues at all. So the first place our investigation is going to take us is to the satellite view, you could say. We're looking at the big picture, using things like brain scans. So what do these macroscopic images actually show us?
So, what these scans are revealing isn't one single major issue. It's more like a whole pattern of subtle but consistent differences spread all over the brain. For instance, you see key areas involved in emotion and memory, like the hippocampus and the amygdala. They tend to be just a little bit smaller. The cortex, which is like the brain's main command center, can be a bit thinner. And the white matter, which is all the wiring connecting different brain regions, it often shows signs of being, well, a little less organized. It's a great analogy. It's not like one bridge is totally out. It's more like the entire city's infrastructure is just slightly less efficient.
And right away, this leads us to the absolute million-dollar question for every researcher in this field. Is this a chicken or the egg situation? Are these brain changes the cause of the illness, or are they the effect? Were people born with these differences, making them more vulnerable from the get-go? Or does the experience of living with bipolar disorder, the intense mood episodes, does that actually change the brain over time?
So this basically breaks down into two competing theories. On one hand, you've got the neuroprogression hypothesis. This idea suggests that the brain changes accumulate over time as a direct result of the illness. But then there's the developmental risk theory, which completely flips that around. It says, no, these brain differences were there from the very beginning, creating a sort of pre-existing vulnerability. The question is it a scar from the battle, or was it a weak spot in the armor all along?
And as if this puzzle wasn't hard enough, here's where it gets really tricky. The treatments themselves can throw a massive wrench in the works. Take lithium, for example, a real cornerstone medication. It actually seems to increase brain volume. It might even be reversing some of these changes we see. But then other medications might do the opposite, or nothing at all. So you can see how this makes it almost impossible for scientists to separate what's caused by the illness and what's caused by the treatment.
All right, so we've looked at the big picture, the satellite view. Now it's time to zoom way in. We're talking about putting on the lab coats and getting out the microscopes. The next batch of clues comes from looking at brain tissue after death, really searching for changes at the cellular and molecular level. So what do we find when we get that close?
One of the key things scientists look for is a process called gliosis. Now, if you think of your brain's neurons as the rock stars, then the glial cells are their roadies. They're the essential support crew. They provide nutrients, they clean up waste, they just make sure the whole show can go on. Well, gliosis basically means the road crew is in trouble. The glial cells are smaller, there are fewer of them, and that leaves the star performers, the neurons, without the critical support they absolutely need to function.
So when you look at all the studies, what does the evidence actually show? Well, we do see these patterns, fewer support cells, even fewer neurons in some of those key emotional regulation centers. We also see hints of neuroinflammation. But, and this is a huge but, the findings are kind of all over the map. Seriously, one study will find something in this specific brain region, but then the next study finds it somewhere else, and a third study might not find it at all. This high variability, this inconsistency, is a massive roadblock. It makes it really hard to draw any firm conclusions.
Okay, so we've got clues from the big picture brain scans, and we've got clues from under the microscope. The really important next step for any good detective is to start connecting those dots. How in the world can these tiny, tiny changes at the cellular level possibly lead to the large-scale differences that actually show up on an MRI scan?
Well, the leading theory is that it's like a cascade effect. Think of a line of dominoes. The very first domino to fall is molecular. Tiny things like inflammation or oxidative stress kick things off. That first domino knocks over the next one, cellular changes. This is where neurons and their support crew start to get damaged or even die off. This, in turn, knocks over the third domino, microscopic structural changes. You can think of it like the forest getting thinner, tree by tree. And finally, when enough of those microscopic changes build up, the last domino falls. The damage becomes large enough to actually be visible on a brain scan. It's this really elegant model that connects the invisible world of molecules to the visible world of brain imaging.
All right, so we've looked at all this evidence. We've gone from the really big picture all the way down to the cellular level. So, where does that leave us? What's the current verdict in our investigation? What do we actually know for sure?
Well, this quote from a recent major review really sums up the bottom line. It says that, at present, no imaging or neuropathological finding has been identified that holds any real clinical or diagnostic significance. And that's a pretty sobering statement. What it means is that after all this work, after all this searching, right now, there isn't a single finding, not from a brain scan, not from under a microscope, that is consistent enough to actually be used in a clinic to help diagnose someone.
Now, to be really clear, that does not mean we found nothing. Not at all. We consistently see changes in the exact brain regions you'd expect, the ones that control mood, impulsivity, and thinking. All the clues are pointing to some kind of fundamental problem with the brain's ability to regulate itself, to maintain what scientists call homeostasis. The problem is just that the findings are too messy and nonspecific to be used as a diagnostic test. It's like we've definitely found the right neighborhood, but we still can't find the exact house number.
And that leaves us with the final big question, the one that really drives this entire field of research forward. How do we crack this code? We know the clues are there, scattered across all these different levels, molecular, cellular, structural. Finding the answer, the thing that ties it all together, is going to take bigger, more sophisticated studies, and probably technologies we haven't even invented yet. But figuring this out, that is the absolute key. It's the key to unlocking better diagnostics, more targeted treatments, and finally, a real, deep understanding of the bipolar brain. The search for sure continues.